All right, welcome to Radon's Smart Review, and buckle up because we're diving straight into the fascinating and, well, clinically crucial world of chromosome damage and repair.
Absolutely. It's fundamental stuff.
With me is our expert in radiobiology, and today our mission is to unpack how radiation, messes with our fundamental genetic blueprint.
Right. What actually happens at the chromosome level?
Exactly, and what the consequences are, and maybe what happens when our cellular repair teams kind of drop the ball.
And we'll definitely connect that to cancer and how we treat it with radiation.
Perfect. So where do we start?
I think the first thing to really wrap our heads around is the difference between chromosome aberrations and chromatid aberrations.
Okay. Chromosome versus chromatid.
Yeah, and this difference, it really hinges on one critical factor when the radiation hits the cell in its life cycle, specifically relative to DNA replication, the S phase.
Okay, let's unpack this. So before S phase, before the cell copies its DNA, the chromosome is just a single strand, right? A single chromatid.
That's right, in G0, G1, or maybe very early S phase.
So if radiation causes a break then, what happens next?
Well, the cell doesn't know the break is there, necessarily. So when it goes into S phase to replicate its DNA, it just copies the damage right along with the intact strand.
Ah, okay. So both copies end up with the same flaw.
Exactly. So when that chromosome condenses later on, getting ready for division, both sister chromatids will carry that identical damage. We call these chromosome aberrations.
Got it. You had that analogy before, like a typo on the master document.
Precisely. You make a mistake on the original, every single copy you make afterwards has that same mistake.
Okay, that makes sense. So what if the radiation hits later, after the DNA is already copied, like late SG2, or even during mitosis?
Right. And now the chromosome already exists as two sister chromatids joined together. If a radiation event causes a break now, it's much more likely to just hit one of those two chromatids.
So only one arm of the duplicated chromosome gets damaged.
Exactly. The other sister chromatid is usually fine. So when you look at it during mitosis, you only see the damage on one side. These are chromatid aberrations.
So that's like spilling coffee on just one photocopy, not the original master recipe.
Perfect analogy. The original, or in this case, the other chromatid, is unaffected.
Okay. Now, I imagine not all this damage is equally bad for the cell, right? Some must be worse than others?
Definitely. Some types of these chromosome changes are much, much more likely to kill the cell. We often call these the lethal aberrations.
The lethal ones, like Dicentrix rings and anaphase bridges.
Those are the main ones, yes. They're often called unstable or asymmetrical aberrations. And they're lethal usually because they cause a big loss of genetic material, or they just completely mess up the process of cell division, mitosis.
Okay, let's tackle Dicentrix first. Sounds dramatic. Dicentric. Two centromeres. How do those even form?
Right. So dicentrics come from that first type of damage we talked about, chromosome type damage, before DNA replication, usually GO or G1.
Okay.
And you need breaks to happen in two different chromosomes. Now, DNA likes to repair itself, but sometimes those broken ends get, well, sticky. And they can rejoin incorrectly.
So a piece from one chromosome hooks up with a piece from another.
Exactly. But specifically, a fragment containing the centromere from one chromosome joins with a centromere containing fragment from the other broken chromosome.
And boom, you've got one chromosome with two centromeres. I can see how that would cause chaos during cell division.
Total chaos, because almost always when you form a dicentric, you also create these leftover pieces without centromeres. We call them eccentric fragments.
Okay, acentric, no centromere.
Right. And during mitosis, the cell uses spindle fibers that attach to the centromeres to pull the chromosomes apart. These eccentric fragments, they can't attach, so they just get lost, left behind.
Bad news. What about the dicentric itself? With two centromeres.
It tries to attach to spindles from both sides of the dividing cell, so it gets pulled in opposite directions. It often forms this physical bridge stretching across the middle of the cell.
Ouch. That can't end well.
It usually breaks, randomly, which leads to more genetic loss, instability, and, yeah, usually cell death. Finding dicentrics is a classic sign of radiation exposure.
Clinically important then. And I think I remember something about the number of dicentrics relating to the dose of radiation.
Yes, absolutely. The yield, or the frequency, of dicentrics follows what we call a linear quadratic model. So yield equals alpha D plus beta D squared.
Alpha D plus beta D squared. What does that mean in simple terms?
Well, the linear part, the alpha D, represents dicentrics formed by a single radiation track causing both breaks needed. Think low LE2 radiation, maybe at lower doses. The quadratic part, beta d squared, represents dicentrics formed by two separate radiation tracks, each causing one break, and those breaks happening to find each other. This becomes more important at higher doses or with higher LET radiation.
So the relationship isn't just a straight line. It curves upwards faster at higher doses.
Exactly. And this relationship is actually super useful. It's the basis for biological dosimetry. If someone's suspected of being exposed in a radiation accident, we can take a blood sample, look at their lymphocytes, count the dicentrics.
And estimate the dose they received. Wow.
Yeah, it's a powerful tool, especially for recent exposures.
Okay, that's fascinating. Let's move on to the next lethal one. Ring chromosomes. They sound circular.
They are. Rings also come from chromosome-type damage, so before replication. But this time you need two breaks on the same chromosome.
Two breaks, same chromosome, one on each arm.
Typically, yes. One break in the short arm, one in the long arm. Then, the main piece of the chromosome, the part between the breaks that includes the centromere, rejoins end-to-end, forming a circle, a ring.
And the bits that broke off, the tips of the arms.
Those become eccentric fragments, just like with dicentrics, and they get lost.
Okay, so lost genetic material again. But why is the ring structure itself so bad?
The ring shape itself causes problems. It can get tangled during DNA replication. It's hard for the cell to properly pull sister chromatids apart if they're rings during mitosis. It often leads to breaks, more instability, and usually cell death.
Are rings as common as dicentrics?
Generally, no. Dicentrics are usually more frequent. Getting those two specific breaks on the same chromosome for a ring is maybe less probable than getting breaks on two different chromosomes that happen to be nearby.
Makes sense. Okay, last one in the lethal trio. Anaphase bridges. Now, these were different, right? Damage after replication.
Precisely. Anaphase bridges come from chromatid-type damage. So the radiation hits in late SG2 or even M phase after the DNA is copied.
Okay, so we have two sister chromatids already.
Right, and what happens is you get breaks in both sister chromatids at the exact same spot. This is sometimes called an isochromatid deletion.
Both sisters, same location. Then what?
Then the broken ends of the sister chromatids can fuse together. It's called sister union. They incorrectly rejoin with each other.
Ah, okay. So the two sisters are now physically linked where they shouldn't be. What happens when the cell tries to divide in anaphase?
Well, anaphase is when the sister chromatids are supposed to separate and go to opposite poles of the cell. But because they're fused at that point of sister union...
They form a bridge.
Exactly. A bridge of chromatin stretches between the separating groups of chromosomes. Just like the dicentric bridge, this prevents proper separation. You also usually get an eccentric chromatid fragment from the initial break, which gets lost.
And does the bridge break?
Usually, yes. The tension causes it to snap, again leading to unequal distribution of genetic material, instability, and often cell death. Like rings, anaphase bridges are generally seen less often than dicentrics.
Okay, that clarifies the lethal types really well. Dicentrics, rings, anaphase bridges. Maybe in the mic. How about DRA lethal? Dicentrics, rings, anaphase bridges are lethal.
Hey, I like that. DRA lethal. That works. Easy to remember.
Great. Now, you mentioned these are the lethal ones. That implies there are non-lethal types.
That's right. There's a whole other category of aberrations that are generally considered non-lethal or stable. We often call these symmetrical rearrangements.
Stable. Symmetrical, meaning the cell can survive these changes.
Often, yes. They can survive and divide. But, and this is a big but, these stable aberrations can have really significant long-term consequences.
Like cancer.
Exactly. They can play a major role in carcinogenesis down the line. The main types we think about here are symmetric translocations, interstitial deletions, and inversions.
Okay, let's start with symmetric translocations. How are these different from the dicentric forming ones?
Okay, so like dicentrics, they start with breaks in two different chromosomes before replication, chromosome-type damage.
Right.
But the key difference is how they rejoin. In a symmetric translocation, there's a reciprocal exchange. A piece breaks off chromosome A and swaps places with a piece that broke off chromosome B.
Ah, a swap. So each chromosome gets a bit of the other one.
Precisely. So you end up with two rearranged chromosomes, but critically, there's usually no significant net loss or gain of genetic material, and each rearranged chromosome still has just one centromere.
So the cell can handle mitosis okay?
Generally, yes. Because it's a balanced exchange, the cell can often divide pretty normally. That's why they're stable and non-lethal in the short term.
But you said they can still cause problems.
Absolutely. Even though the amount of DNA is right, the rearrangement itself can be damaging. A breakpoint might happen right in the middle of an important gene, breaking it.
Disrupting the gene function.
Exactly. Or The translocation might move a gene to a new location where it's controlled differently. Imagine moving a growth-promoting gene, a proto-oncogene, next to a really strong on switch, like an enhancer element that's always active.
That sounds like a recipe for uncontrolled growth.
Yeah.
You mentioned examples, CML and Burkitt's lymphoma.
Classic examples. In chronic myelogenous leukemia, CML, you get a translocation between chromosome 9 and 22T922. This creates the famous Philadelphia chromosome.
Right.
What happens is the ABL gene from chromosome 9 fuses with the BCR gene on chromosome 22. This creates a new fusion protein, BCR-ABL, which is basically a permanently on growth signal, a tyrosine kinase, driving the leukemia.
Wow. And Burkitt's lymphoma.
There, it's often a translocation between chromosome 8 and 14, T814. This puts the MYC gene, a powerful proto-oncogene on chromosome 8, right next to the immunoglobulin heavy chain enhancer on chromosome 14, which is super active in B cells.
So MYC gets massively overexpressed.
Exactly. Driving the lymphoma. So these stable translocations can be initiating events for cancer years later.
And because they're stable, can they stick around for a long time?
Yes, that's another key point. Especially in long-lived cells like lymphocytes, these symmetric translocations can persist for years, even decades, after the initial radiation exposure.
Which has implications for that biological dosimetry again.
Right. While decentrics are good for recent exposure, looking for stable translocations using more sensitive techniques like FIJ can give us an estimate of radiation dose received much further back in time.
Okay, FIC, so we'll get to detection methods. What about interstitial deletions? You mentioned deletions with rings, but these are different.
Yeah, interstitial deletions also start with chromosome type damage. 2 breaks again, but this time both breaks are on the same chromosome arm.
Okay, 2 breaks, same arm.
The piece of the chromosome between the 2 breaks gets cut out and lost. The 2 broken ends on the main chromosome piece then rejoin.
So you end up with a chromosome that's just shorter, missing a chunk from the middle.
Exactly. It's lost an internal segment. Whether this is lethal or not really depends on how big that chunk is and what genes were in it.
Makes sense. Small deletion might be okay. Big one might lose essential genes.
Right. And critically, for cancer, if that lost segment contained a tumor suppressor gene.
You've lost a break on cell growth.
Precisely. That can contribute to cancer development too.
Okay. And the last stable type was inversions, flipping a segment.
Correct. Again, 2 breaks on the same chromosome, but instead of the middle piece getting lost, it gets flipped 180 degrees and then rejoined back into the chromosome at the same spot.
So the gene order in that section is reversed.
Exactly. There's no net loss or gain of material, so these are generally non-lethal, balanced rearrangements.
But can they still cause trouble?
They can. If one of the breaks happens within a gene, it can disrupt it. Or even if no gene is broken, changing the order might affect how nearby genes are regulated. So while generally stable, they're not always completely harmless.
We've covered a lot of ground on the types of damage. How do we actually see these things? You mentioned FESH.
Right. Well, the classic method is karyotyping. You basically grow cells, stop them in metaphase when chromosomes are condensed.
Squish them onto a slide.
Pretty much. Then you stain them, usually with GEMSA banding, which creates a unique pattern of light and dark bands on each chromosome. You look under a microscope, arrange them by size and banding pattern.
And look for obvious changes, like a whole missing chromosome, or really big deletions or translocations.
Exactly. It's good for large-scale changes.
Yeah.
But it has limits. It might miss subtle rearrangements.
So that's where FESH comes in, fluorescence in situ hybridization.
Yes. FAH is much more specific. You design these small DNA probes that are tagged with fluorescent molecules. These probes are designed to bind only to specific DNA sequences you're interested in.
So you could have a probe for a specific gene or a whole chromosome.
Both. You can use probes for specific gene locations to see if they're deleted or amplified, or you can use chromosome painting probes, where a whole chromosome is labeled with a specific color. This makes it much easier to spot translocations because you'll see, say, a piece of red chromosome stuck onto a green one.
Ah, okay. Very visual and useful for confirming dicentrics, too.
Definitely. You can use probes that stick to the centromeres. A dicentric will light up with 2 centromere signals.
And then there are even fancier versions, SKY and MFISH.
Right. Spectral karyotyping, SKY, and multicolor FESH, MFIs. These use combinations of different fluorescent dyes to give each of the 23 pairs of human chromosomes a unique spectral signature or color.
So you can see the entire chromosome set painted in different colors all at once.
Exactly. It's incredibly powerful for visualizing very complex rearrangements where maybe 3 or 4 different chromosomes are all involved in swaps and breaks, which you often see in cancer cells. It gives you a complete overview of the chromosomal chaos.
So tying this all together, these aberrations have huge implications, right? Both for killing cancer cells and maybe for causing cancer later on.
Absolutely central. Those lethal aberrations we started with, dicentrics, rings, anaphase bridges, their induction, is a major reason why radiation therapy actually works. It causes so much damage that the cancer cells simply can't survive division.
And the frequency of these lethal hits correlates with cell kill.
Directly. More lethal aberrations, more cell death. That's what we're aiming for in the tumor.
But the flip side is the stable, non-lethal ones, translocations, deletions.
Right. While they don't kill the cell immediately, they can cause those long-term problems we talked about, like activating oncogenes or knocking out tumor suppressors. That's a mechanism for radiation potentially inducing secondary cancers years down the line.
And we also use them for that biodosimetry, tracking exposure.
Correct. Dicentrics for the short term, telling us about a recent exposure. Translocations for the long term, sticking around for years, telling us about past exposures.
This really highlights how complex the response to radiation is. And it must vary between people, right? Especially if their basic DNA repair machinery isn't working properly.
Precisely. This leads us right into the topic of DNA repair deficiency syndromes. There are several inherited genetic disorders where people have mutations in genes that are crucial for spotting or fixing DNA damage, especially those dangerous double-strand breaks that radiation causes.
And these syndromes often mean more chromosome problems, higher cancer risk, maybe different sensitivity to radiation.
All of the above. These individuals often show increased baseline levels of chromosome aberrations. They have a much higher lifetime risk of certain cancers. And critically for us, they can be significantly more sensitive to the effects of ionizing radiation.
Okay, let's go through some key ones. Ataxia-telangiectasia, AT, that's a big one, isn't it?
AT is probably the classic example of a radiosensitivity syndrome. It's caused by mutations in the ATM gene.
ATM, what does that do?
ATM is like a master controller. It's a kinase that senses DNA double-strand breaks and kicks off a whole cascade of responses, activating repair pathways, halting the cell cycle to allow time for repair. It's absolutely critical.
So if ATM is broken,
the cell can't respond properly to double-strand breaks. Patients with AT have progressive cerebellar ataxia problems with coordination, those little dilated blood vessels called telangiectasias, often in the eyes and skin, immune deficiencies, and a very high risk of lymphomas and leukemias.
and their response to radiation.
Extreme radiosensitivity. Their cells are incredibly sensitive. We talk about the D0, the dose to kill about 63% of cells. For AT cells, it's maybe around 0.5 gray, whereas for normal cells, it's usually 1 to 1.5 gray.
Wow, so a tiny dose does huge damage.
Exactly, which means standard radiation therapy doses can be catastrophic for these patients. It's often contraindicated or requires massive dose reductions and extreme caution.
Crucial to know. What about similar sounding ones like Nijmegen breakage syndrome, NBS?
NBS is another important one. The gene here is NBN, which codes for a protein called nibrin, or NBS1.
Nibrin.
Nibrin is part of a complex called mRN, mRE11, RAD50, NBS1. This mRN complex is one of the very first things to arrive at a double-strand break. It helps process the broken ends and also helps activate ATM.
So a problem here also messes up the double-strand break response.
Right. Patients with MBS often have microcephaly, small head size, growth problems, immune deficiency, high risk of lymphomas, and yes, they are also radiosensitive, though typically not quite as extreme as AT.
And there's ataxia-telangiectasia, like disorder ATLD.
ATLD is caused by mutations in a different part of that same mRN complex, the mRE11A gene. So again, problems with sensing and processing double-strand breaks and activating ATM. Clinically, it's often a bit milder than AT, maybe less severe neurological issues, but still immunodeficiency, cancer risk, and radiosensitivity.
Okay, so ATM and the mRN complex, MBS, ATLD, are all key for double-strand break response and radiosensitivity. What about Fanconi anemia, FA? That involves lots of genes.
Fanconi anemia is complex. There are over 20 different FANC genes identified now. They form a pathway that's really critical for repairing a specific type of damage called DNA interstrand crosslinks, where the two strands of DNA get stuck together.
Crosslinks. Like from chemotherapy drugs.
Exactly, like mitomycin C or cisplatin. FA cells are hypersensitive to those agents, but the FA pathway also interacts with pathways that repair double-strand breaks, particularly homologous recombination.
So it affects radiation response too.
Yes, patients with FA have developmental issues, bone marrow failure, high risk of leukemia, and certain solid tumors, and they do show moderate radiosensitivity. Not usually as severe as AT, but definitely increased sensitivity.
Okay, then there are syndromes involving helicases. Bloom, Werner, Rothman-Thompson.
Right. These involve mutations in genes like BLM, WRN, RECQL4, which code for RECQ helicases. Helicases are enzymes that unwind DNA, which is important for replication, repair, recombination, basically keeping the genome stable.
So defects cause instability.
Yes. These syndromes feature things like growth problems, sun sensitivity, Bloom, Rothman-Thompson, signs of premature aging, Werner, genomic instability, and higher cancer risk. Radiosensitivity can be a feature, but it's often more variable than in AT or NBS.
Now, are there repair defects that don't cause significant radiosensitivity? You mentioned some earlier.
Good point, yes. Lynch syndrome, for example, also called HNPCC.
Hereditary non-polyposis colorectal cancer.
Right, that's caused by defects in mismatch repair genes, MSH2, MLH1, etc. These fix errors made during DNA replication. Crucial for preventing mutations that lead to cancer, especially colorectal and endometrial cancer, but mismatch repair isn't a major player in fixing radiation-induced double-strand breaks. So Lynch patients aren't typically considered radiosensitive.
And xeroderma pigmentosum, XP.
XP is the classic example of UV sensitivity. The defect is in nucleotide excision repair, NER, which fixes bulky DNA damage caused by UV light. XP patients have extreme sun sensitivity and sky-high rates of skin cancer. But again, NER isn't the main pathway for fixing ionizing radiation damage, so they generally have normal radiosensitivity.
Interesting distinction. What about BRCA1 and BRCA2? They're huge in breast and ovarian cancer, and they are involved in double-strand break repair, right? Homologous recombination.
Absolutely. BRCA1 and BRCA2 are critical for high-fidelity repair of double-strand breaks using that homologous recombination pathway. Mutations dramatically increase risk for breast, ovarian, prostate, pancreatic cancers.
So are BRCA carriers radiosensitive?
That's a more complicated question. There is definitely laboratory evidence that BRCA-deficient cells can be more sensitive to radiation because their HR repair is faulty, and some clinical studies suggest maybe slightly increased normal tissue toxicity in carriers. However, it's not nearly as profound or consistent as something like AT. It's still an active area of research to understand the full clinical implications for radiotherapy.
But their tumors can be sensitive to certain drugs because of that defect.
Exactly. That's the basis for PRP inhibitors. PRP enzymes are involved in other repair pathways, like single-strand break repair. If you inhibit PRP in a cell that already lacks BRCA function, and thus good HR repair, the cell accumulates too much damage and dies. It's called synthetic lethality. Also, why these tumors can be more sensitive to platinum chemotherapy, which also causes damage, HR helps repair.
Okay. This really ties together DNA repair, cancer risk, and treatment response. It's incredibly complex.
It really is. Maybe that analogy helps.
Yeah.
Think of DNA repair like the cell's construction and maintenance crew.
Yeah.
Radiation is like causing damage, breaking walls, pipes. The repair pathways are the different specialist crews, electricians, plumbers, framers that fix things.
Right.
In these syndromes like AT, it's like the main foreman, ATM, who coordinates the double-strand break repairs is missing or faulty. Or maybe the specialized crew for crosslinks, FA pathway, is short-staffed.
So repairs don't happen correctly, or they're slow, leading to more persistent damage, those chromosome aberrations.
Exactly. And a higher risk of the whole structure becoming unstable or collapsing, leading to cancer. And it also means the structure is more vulnerable to further damage, like from radiation therapy.
That's a great analogy. It really helps picture how interconnected it all is.
Glad it helps.
All right, let's maybe crystallize some of this with some clinical pearls. What are the absolute key takeaways?
Okay, pearl number 1. Remember the timing. Damage before S phase means chromosome aberrations affecting both chromatids. Damage after S phase means chromatid aberrations affecting just one. That's fundamental.
Got it. Timing is key. Pearl 2.
Lethal aberrations, DRA, lethal, dicentrix rings, anaphase bridges. They kill cells primarily by causing massive loss of genetic material or by physically disrupting mitosis. That's how radiation kills tumor cells.
Okay. Lethal means mitotic catastrophe or major loss.
Yeah.
Pearl 3.
The non-lethal, stable aberrations, especially symmetric translocations and deletions, they can stick around. They might not kill the cell now, but they can cause cancer later by messing with oncogenes or tumor suppressors. And they're useful for that long-term biodosimetry.
Excellent summary. Ready for a quick board blitz? Test ourselves.
Let's do it. Fire away.
Question 1. You observe a chromosome preparation at metaphase. You see an aberration that clearly has 2 centromeres, and there's also a fragment floating nearby with no centromere. What is this most likely? A. Symmetric translocation. B. Ring chromosome. C. Dicentric chromosome. D. Anaphase bridge.
2 centromeres and an eccentric fragment. That's the classic description of a C decentric chromosome.
Correct. Question 2. A patient presents with progressive cerebellar ataxia, has those little blood vessels visible on the eyes and skin, oculocutaneous telangiectasias, and has recurrent infections due to immunodeficiency. Genetic testing reveals a mutation in the ATM gene. What response would you expect their cells to show to ionizing radiation? A. Normal radiosensitivity. B. Increased resistance. C. Sensitivity only to UV. D. Extreme sensitivity to ionizing radiation.
Ataxia telangiectasias, ATM mutation, that's ataxia telangiectasia, or AT. They have D, extreme sensitivity to ionizing radiation.
Exactly. Question 3. Which type of chromosomal aberration is known for being generally non-lethal, stable enough to persist in lymphocytes for many years after radiation exposure, making it useful for biological dosimetry long after the event? A. Dicentric chromosome. B. Ring chromosome. C. Eccentric fragment. D. Symmetric translocation.
Okay, non-lethal, persists for years, used for long-term biodosimetry. That would be D, symmetric translocation.
Perfect. Last one, question 4. If radiation causes DNA damage during the G2 phase of the cell cycle, what type of aberration would you most likely expect to see when the cell reaches mitosis? A. Dicentric chromosome. B. Ring chromosome. C. Symmetric translocation. D. Chromatid aberration like an anaphase bridge.
G2 phase, that's after DNA replication. So the damage would affect individual chromatids. That points to D, chromatid aberration. Anaphase bridges are a type of chromatid aberration.
Nicely done, 4 for 4. So just to wrap it all up then, we've seen how radiation breaks DNA, leading to these different chromosome problems.
Right, and whether it's chromosome type or chromatid type depends entirely on when in the cell cycle that break happens relative to DNA copying.
The lethal ones, dicentrics, rings, bridges, are bad news immediately, causing cell death, which is good for killing tumors, but bad for normal tissue.
While the stable ones, translocations, inversions, some deletions can survive but might lead to cancer down the road, and we can use them to measure past radiation doses.
And finally, understanding those inherited DNA repair defects, like an AT, MBS, is just so critical because they tell us who might be at higher risk for cancer and who might react very differently, often much more severely, to radiation therapy.
Absolutely. It touches everything from basic radiobiology to tumor control probability, normal tissue complication probability, and even patient selection and counseling. It's core knowledge for radiation oncology.
Couldn't agree more. A really fundamental topic.
Definitely.
Well, that's all the time we have for today's deep dive. A huge thank you for walking us through that, and thanks to all of you for tuning in to Rad on Smart Review.